All right, welcome everyone. My name is Grace Ann Taylor. I'm the Education and Outreach Coordinator at Save Barnegat Bay. And tonight we have the pleasure of welcoming Kent Mountford to speak about his work with plankton in the past here in Barnegat Bay. He's also the author of a book called Closed Sea, which we have a video of his Meet the Author event on our YouTube channel, on Save Barnegat Bay's YouTube channel. And it was our first Meet the Author event, so we're bringing it first full circle to invite Kent back to speak about the science that he did, not just as a writer, but as a scientist, to talk about plankton. So I'll let you take it away.
Thank you very much. Pleasure to be here. And thank you all for attending. Sorry about the mix-up the last time. I was ready to go. The internet link was not. Sorry about that. This is a pretentious title that you could... salvation of the world would be plankton. It could be true, and I'll try to make that argument a couple of times during this presentation. But this really all started in Barnegat Bay, but it started a long time ago. And if I can figure out how to make my next change on this, here we go. I'm going to talk to you about various kinds of plankton. The groups, major groups of plankton that we'll be discussing today are the diatoms, the dinoflagellates. The pictures are on the left, top row on down. Microflagellates, very, very tiny little critters, and nanoplankton, or now called picoplankton, and some important effects of these. That is a fish skeleton in the bottom left, and that's one of the effects that plankton can give you. This started actually back for me in 1961 when I ended up as a Army recruit at Fort Dix. And this was not a time to be a relatively new college graduate because no one would hire you. And when I got out of the basic training problem that I put through my 6 months in active duty, no one would hire me. I was struggling for a job. The result was that I didn't want to waste the time. I spent an awful lot of time in this old cottage at the left, which is on Manasquan Beach. It looks right at the ocean. And I spent an awful lot of time tramping the beaches, tramping the pine barrens, and learning what I could about the world I lived in. And that was really a wonderful time for me. I'll never regret having that free time. I went a lot to Island Beach, and this kept me in touch with Barnegat Bay. I was between boats at the time, and it also gave me an opportunity to walk both the shores of the bay and the shores of the Atlantic Ocean. I did this at all hours of the day and night, and one of the things I encountered was luminescence in the surf at night. That was just really exciting to me. And I found that if you went down to the beach, you could kick the sand, and there would be luminescence there as well. And I don't know how many of you have ever seen this, but it's really a cool thing. I decided I'd try to measure this and figure out what was actually happening, maybe figure out where it came from. So I created what's called a comparator, this little sheet. This is the original little piece that I wrote. And when there were just a few dots, I would mark, number 1, and I would go on up until it was almost solid, beautiful luminescence in the sand. I had no idea what was doing. A year passed, and I was gathering this data, and I finally got to a seminar up in the Sandy Hook Marine Lab. One of the scientists sent me an email, actually through the American Littoral Society. A woman who was then head of the American Littoral Society put me in touch with this guy. And so I went to this seminar, and I realized that the luminescence was coming from plankton, and particular kinds of plankton, to be sure. So I went out to what was then a Lafayette Radio and Electronics Store. Probably 3 or 4 of you were alive when that chain was in business. It was a predecessor of Radio Shack, etc. In 1963, I bought a kid's microscope for $4 with a little dissecting kit that went along with it. And I began to look at what was going on. And I thought, well, I got to get something out of the water. So I heard about plankton nets. That's my mother's used nylon stocking on the left. And that's where I started pulling plankton out of the sea. The toe was much better than the other part of the weave. It was too big and it let an awful lot of plankton through. It took me a while to learn about that. Eventually, I went back to graduate school, and I had real plankton nets like the one on the left, which is designed to allow you to calibrate how much water went through the net. And I carried that thing literally halfway across the world. This is actually in the Caribbean on the island of St. John, and I did it in Scandinavia and in Germany, and ultimately many, many places in the world. And as a graduate student, I began to study plankton seriously. I got a decent microscope and a small research grant. It was the time when Barnegat Bay was about to be attacked by the Oyster Creek nuclear power plant. And that was the reason that I was able to fund my work. And of course, plankton is dependent upon photosynthesis. It's really the energy of the sun being translated into organic matter that's useful on the planet Earth. And the sun's energy is really foundational to almost all life on Earth. There are sidetracks and decomposition products that also work with living organisms and produce energy sources of different nature. But it all goes back to the sun. You can't really avoid that. Very important thing in the world. I began to study plankton, and I learned many of the species through the microscope and through various pieces of literature. And I began to track what they were doing at various seasons of the year and continued that for most of 4 years, which you see here, and then went on in 1971 when the nuclear power plant was in operation. And my dissertation was to look at how the plant affected the plankton in Barnegat Bay. Well, you'll notice these sort of dashes and whatnot. They really run in 4 patterns that each of the 4 years repeated what was happening in the previous year in many ways. There were organisms that didn't show up, organisms that did. But this pattern really was extremely important in understanding how the plankton cycled through the year, largely driven by temperature, but also driven by many ecological factors that we'll discuss. When it's cold and the sun gets strong and stronger and stronger and stronger, it's also relatively wet. We had a very wet spring this year, so you had an example of how a wet spring increases river flow and groundwater flow from the land into Barnegat Bay. That was very important to the plankton, and this occurred all along the coast. It occurs in every year to a different degree. This particular group of plankton, the plant plankton, are fed by the sun, and they are fed by the rivers, fed in terms of food by the rivers and the land runoff themselves. They are extraordinary organisms, among the most beautiful things on the face of the earth. There are a few of them shown in this picture, but they are fed by nutrients, in our case, from the forest, from the marshes, and also from the air in terms of nitrogen, particularly that's brought down by rainfall. And particularly in the pine barrens, which are very sandy terrain, the mineral silica is actually dissolved from the sand itself, and it comes into the river in all our rivers and all our streams and the groundwater as well, as a mineral content of the water from sandy soil. In the far past, let's go back a couple of centuries, in the 16th, 17th, 18th, 19th century, the nutrients leaching from those pine barrens were really very, very small. And the plankton would run out of silica and out of nitrogen as well. And the salt marshes also were actually releasing some nutrients, but in many ways they retained nutrients very well. That's how they grew. They were fertilized by stuff from the land as well. The pine barren rivers used to run out of nutrients annually, and plankton growth generally slowed in the summer. I've been around the bay and around this estuary long enough that I go back to information that was gathered in the late 1950s and early 60s by a guy named Jim Durand, who was a professor at Rutgers down in Camden. And he studied primarily the Molokai River in Little Lake Harbor, which is now sort of part of what we call the Barnegat region. And those rivers, he found, ran out of nutrients. They really reduced nutrients to the point where both silica and nitrogen limited the growth of plankton. So you had plankton growing much more slowly in the summer And the rivers, the bays and the coastal ocean got clearer after that very strong growth in the early spring. So that's something which no longer occurs in Barnegat Bay. Between the parkway and, atmospheric and impervious surface runoff, the loss of marshes, and the development in general, we spoiled all that. And I think all of you are here because you understand that, and that's really why Barnegat is being looked at in terms of both research and citizen activism. The fertilizer that was never in the Pine Barrens is now in Barnegat Bay. And I thank Woolley for really doing some of the early homework on that and getting some kind of legislation that tried to make a dent in that. This has served as a model in many other estuary programs, including the one in Chesapeake where I worked for many, many years. Too many. But that's a key thing. Plankton don't give a damn where they get the nutrients from. If you feed them in any way with all the different things that they need, both organic molecules and minerals that are in the water and atmospheric inputs and the stuff we dump in from garbage dumps and whatever, they will grow. And are not always the kind of plankton that we would like to have. So I'll talk about diatoms first. These are plants. They rely on photosynthesis to build their cellular material. And all of them build glass-like capsules of silica, which is why they need the silica, around their living parts. The sculpture of these things is just remarkable. They're such beautiful things. This is just the tip of a gyrosigma cell. I may have a picture of it later on, we'll see. But look at the detail on that. That little, almost like plaid or crisscross or screen-like pattern in the cell. Just amazingly beautiful. And they're almost endless in their variety and form. The one on the left is called Coconus. It's a diatom that grows in nearshore shallow waters. The middle one is Bidulphia, and it's an open ocean diatom that shows up here kind of rarely, particularly in the spring. And the one on the right is Chitoceras, and it forms longer chains, about which I'll talk in a couple of minutes. There are literally thousands of kinds of diatoms, and really remarkable critters. This is one of them. The one on the left, I will let out a secret. It is one that stuck to the wall of my garage with the sea foam that came in when my house was destroyed by Hurricane Sandy. And it was there reminding me that plankton was in the water that destroyed my home, which is 100 years old at that time. A close-up of the edge of that cell shows these tiny, tiny pores and patterns in this glass structure that the diatom built. This is the dome of the Mosque of Sheikh Lutafallah Istafan from the 1600s. It's an illustration from the British Museum of how similar that is to the diatom, which was created by nature millennia before. In my well here in southern Maryland, we had a failing well, and it brought up ancient sediments with the water and messed up our whole water system. One of the diatoms was that tuxin hibiscus, which you saw in the last picture. And so they've been around for tens of millions of years. Now, these animals have a role in the ecosystem as well, and they're eaten by animal plankton, which is called generally zooplankton, the Latin word for animal. And this is a copepod, which means four-footed. These are two big antennas at the top. She will swim, sweeping them back and forth and zip through the water in a jerky pattern. And she's about 100 times the size of a diatom. So she can eat those things, and that, of course, is what our fisheries are based upon, this animal plankton. That's a whole other talk. I won't get into much of that. But diatoms grow by forming a new cell inside the old. It's kind of like mitosis in human cells, where one form helps. My internet is unstable. Help! Does that show on your screen? Maybe not. Anyway, my internet is always unstable. So am I.
You're good.
These things form capsules like a pillbox. and the top fits inside the bottom. This is a little cyclotella diatom that's very common in Barnegat Bay. And these forms many times stay attached in a chain. You can see a bunch of cells attached in a long chain here. And in the bottom one, which has died and the cell material has left the cell, you can see where the top and bottom of the capsule actually join. These can be hundreds of cells long, and they sink very slowly. They're like a hair in your soup. And they then drop out of sunlight slowly, and then maybe the waves will bring them back up so they continue photosynthesis. But they're also harder for eating by those other organisms, the zooplankton, because where do you start on a chain that's 100 cells long? You start at one end and start chewing on it. You could eat a long time on a chain of 100 cells. Diatoms form these blooms in February and March before they run low on silica, particularly. And they also are dependent upon colder water. They're much more happy in cold water. So as the spring bloom ends, the diatoms begin to die out. But that's the time when the zooplankton also come in. I had a slide which I couldn't use because it was too faded. I had two tubes from my plankton net. One tube was the diatoms, which were very green in color. The second tube was pinkish from the little animal plankton that occurred just 14 days later in my sampling program. So this shift and the value to the organisms that eat the diatoms is incredibly valuable. In 1906, there was an oyster biologist from Rutgers. He was actually an agricultural biologist, and he taught himself marine biologist. He was the first biologist who really worked in Barnegat Bay. And he said that one of these diatoms, Skeletonema costatum, is the $1 million item because what it did to the oyster industry was feed the oysters abundantly. Oysters loved them. And these organisms are still in Barnegat Bay, and you can see them here. They're two different views of the same critter. Notice the little spines that connect each cell. They're almost like little water towers with legs under them. And each of the dark areas is a living cell, and the spines are what connects them, and the water circulates among them. This is a cell came from Barnegat in the winter of 1968, and they're still there, but they're not the once hundreds of acres of oysters. There are in Delaware Bay, and the diatom is still $1 million diatom in Barnegat, in Delaware Bay, and in Chesapeake Bay as well. This is another of the copepods. This is a female, and she's got a big bunch of eggs hanging on her butt. And you can get the idea. She's about 0.2 of a millimeter long. And it's kind of interesting that up in the upper right, these are drawings I did 57 years ago in 1964. They're the little cyclotellas that I showed you a picture of earlier. And she could crunch them up. Her mouth is on the underside of her body. She only has one eye, but she's out there looking for food. Now, 10 bugs that you find in the surf when you go swimming on the beach, mole crabs, Amareta taltuita, are also feeding on plankton, but they get the plankton that's filtered out in that sand the way I was seeing it when I was a young guy looking at luminescence in the sand. And mole crabs, of course, when they're stirred up by the surf in shallow water, are excellent food for striped bass, the fillets of which you see on the left. Striped bass really can munch up on those crabs. Now, the bay's bottom, Barnegat Bay's bottom, is packed with billions of worms, clams, and larvae. Of course, there's still some harvesting of clams that occur, particularly hard clams. And I don't know if you do soft clams in the Barnegat or not, but there are also millions of larvae that occur from all these organisms. Well, worms and clams and mussels and oysters and, you know, all the commercial species have larvae, little baby parts that come out and float around in the plankton, and that's the critters that are eating the plankton we've been talking about. Over on the right is a face view of a clamworm, Nereus, which is a very common fisherman's bait. And they're also dependent upon the plankton itself and also what settles out on the bottom and is eaten by the critters that are actually roaming around on the surface. Look in the background picture and you see those little tube-like things. Each one of those contains a little animal, and the bottom is full of that stuff. That's my wife's business. She looks at them. and she's done a lot of work in Barnegat Bay. Now, Menhaden also, I'm sure most of you spent time on the water, look at schools in Menhaden, and realize that they also grow up to be an incredibly important commercial species, probably one of the biggest tonnage fisheries on the East Coast and the Gulf Coast. Menhaden devour plankton. They're really set up so that you see the little Menhaden on the right there that's been bitten in half by the bluefish. They're actually designed to filter plankton from the water, just like my mother's nylon stocking. On the left is one of my former student and technicians who has just brought up a net full of Menhaden. He's studying them. And up in the top part is a fishing lure that I caught a bluefish, and he barfed up the poor little Menhaden that he just eaten. So there's your food chain, me at the top in this case. There is one of the diatoms from the stomach of a Menhaden, and it's the same one we've been looking at. There are the Menhaden in the net, which is how they're caught. On the right is one of the products that Menhaden produce. This is fish meal. It's very high in crude protein. I think you can see that where the arrow is pointed. And it's used feeding chicken. And it was a product that came out of Tuckerton in the time when there was a big Menhaden plant in the lower Mullica River. There used to be 22 such plants along the coast that processed Menhaden. There are now 1, and that's in Reedville, Virginia. Now, in summer, the warmth of the water makes plankton very different. As I mentioned, most of the diatoms are dependent upon colder water, and so plankton, when there is enough food for them, become a problem. Now, this is an example of one that most of us who are boaters face. Look at all the barnacles on that rudder, and over on the left is the baby of a barnacle looking at a diatom. And the adult barnacle will feed on that diatom, but the babies can't. They eat other stuff that's very small. Those barnacle larvae, Anopleus is called in Latin, they swim around until they find something like where the bottom pane isn't particularly toxic, and they will settle and become a barnacle. They actually stick their head, that flat portion, to the surface of whatever they're landing on and develop the shell around themselves. They're related to a crab, and they have the same number of plates in their house as a crab does sections in its shell. Now remember that river flow is declining in the summer. You get thunderstorms, of course, but the diatoms are running out of silica, but there's still a lot of nutrients, particularly in today's Barnegat Bay. And one of the reasons is the development that we've allowed to occur. On the right over there is 1956, when I was cruising in my little sailboat. You could pull into that little lovely creek at Stouts Creek and have a wonderful anchorage. There was a little sand beach, and you could go ashore, build a little campfire, cook your dinner, and go back to sleep on your boat. Very wonderful. And that's up top what happened to Stouts Creek. And on the left is what it looks like from the water today. I don't know how anything useful can live in those canals. Sea nettles, I'll bet you they do. This is a little model I made of the lower Barnegat Bay. This was in connection with my work around the nuclear power plant. And you can see on the left that loop that's formed by Forked River, where water was sucked in, and Oyster Creek, where water and heat were pushed out. I had that on the wall of my graduate laboratory, and it's still on the wall of my house in Manasquan. Now, there's a deeper area over against the Island Beach Peninsula where you could come into the shore, and this is the landing at what is now landing A7 in Island Beach State Park. And we had a stove that sailed on my little cruising sailboat. My friend Ben Green and I went ashore. We cooked both our dinner and our breakfast in a campfire on that beach. I don't think you could try that today with any safety. It'll be on you in a minute. Tice's Shoal is where that area is, and excuse me, it is a very, very active boating area today. The picture on the right is nothing compared to what happens there in the summertime. And I have a slide, which isn't mine. I don't feel free to use it, in which there are 300 boats anchored there at Tice's Shoal. Now, I realize some of the efforts of Save Barnegat Bay has involved control of jet skis. It's unfortunate that this couple is my model for a jet ski. They came into A7 Long Island Beach when I came there in my own boat several years ago. But they were a really nice couple, and they operated that jet ski very responsibly. They were at very low speed, didn't damage a single blade of submerged aquatic vegetation. Nice folks, and a proper use of that little high-speed spinner. Now, a second group of phytoplankton I wanted to discuss is the Dinoflagellate. And they can create red tides, or very, very dense blooms, particularly in warmer water. It's been a terrific problem in parts of Florida over the years, and they are also, many of the species are also bioluminescent. In other words, they glow when you bump into them in the water. And the ones on the right is Noctiluca-milaris. I've only seen that one time. This was actually taken at Beaton, and they were brilliantly luminescent, and I was able to actually get them under my microscope and photograph them. The one on the left is a Gonulac species, and it also is a luminescent dinoflagellate. Now notice there's like a groove that's running around this dinoflagellate. I'll give you another example of that. This is one that I found at Beaton's Boatyard in Mandaloking when I was doing my plankton work there. And it has a groove that runs almost all the way around the shell, the actual capsule. These are made of an organic material. They're not made of silica. But that capsule has a groove that's broken in the front, in the middle of that cell on the right. And that's where the flagellum that comes out. It's like a sperm cell that has a flagellum on it. And there are two flagella that come out. One sticks down toward the bottom of the cell, and the other one goes around the groove. And it beats almost like a little whip in that groove. The cell actually rotates as a result of that. So dinoflagellates swim in a spiral pattern, and they're propelled forward and back by the second flagellum that hangs out the back. Now, this is a red tide that occurred at Manasquan in the ocean, not in Barnegat Bay. And up on the right is an example of the two whip-like flagella that these guys use for swimming. These little critters are a porcentrum, and they literally made the water look like tomato soup. I weighed it in, and I took a sample, and this is what it looked like under the microscope. These are not directly harmful, although later on in science, maybe 20 years ago, they were discovered to have some kinds of toxins in them as well. The dinoflagellates sort of act like animals. They can direct where they want to go. They swim up during the daylight for sunlight. If the sunlight gets too bright, they will swim down in the water, and actually, you'll see this in a moment, they will swim down in order to get away from too much light. And then particularly late in the day, they will swim down in the water column to get nitrogen near where the sediments are and where other organic materials have been settling out on the bottom. A generation of these blooms can be so intense that they'll produce gases that will hurt your lungs. I actually experienced this once in New Jersey, and I was surfing and I just coughed and coughed and coughed. I had to leave in my car to get away from the coast in order to get away from this. It's very, very annoying. They will also produce gases that'll discolor house paint. I'll talk about that in a minute. In 1964, there were still wastes coming into Barnegat Bay. These ocean outfalls had not been done then. This is a little sailboat that I had and cruised all over Barnegat Bay. Great joy to me. Look at the one on the right is an outhouse on Island Beach State Park. Wouldn't you love to have that door? It's a museum piece. God knows where it is. I found the outhouse again when I returned. Someone had taken the door. At least I had a picture. So, lovely idea. Now, this is a red tide over on the left in Chesapeake Bay. It was taken by Wolfgang Vogelbein, and it's an aerial view of the Goodwin Islands. If you look in the upper left, you see the vegetation on the island. The water is fairly clear, and the dinoflagellates have migrated down in the water, so they form layers. And where they get the light that they really enjoy, they will stay. And they form these amazing layers, almost look like layers of sediment in the water. But it's all billions and billions and trillions of living organisms. And it was a dinoflagellate bloom. Excess nutrients in the Barnegat Bay caused a red tide in 1964. which brought my sailboat into play. It was the cell that you see in that photograph, Polytrichos, and there were probably 3 species. Listed the other 2 at the bottom of the picture. But the water was soupy in that it actually had a texture to it. It was almost oily, and it smelled fishy. There were baby flounder dead all over the surface of the water, baby sticklebacks that lived in the eelgrass, baby blowfish that had a very abundant breed that year. And the paint on houses in Mantoloking were actually, those painted with white lead paint in those days, were discolored by the hydrogen sulfide gas that was emitted by the decomposition of this plankton. And there were fish kills. This is not a Barnegat fish kill, but there were fish kills all over the place during those blooms and many others. Now, Jen Prager was one of my mentors at Sandy Hook Fisheries Lab, and she said the water looked like tomato soup, so he said, why don't you take your sailboat and go out and look for this bloom? So the bottle on the right, you can see how red it actually was. So I took my little sailboat, and my college roommate and I went down and we looked for the red tide. This is the original paper that we sketched on, held together by old-fashioned Scotch tape. It was celluloid stuff with lousy glue. You can see the blooms that we encountered on Barnegat Bay. And in the center, I have put down the cell counts that I did through my microscope after I came back, and they range from 15,000 to over 9 million. So there are millions and millions of cells in that. And the density of this stuff actually took oxygen out of the water. It did not have the technology to measure it then, but the oxygen was probably the main reason that all the fish were dying. So we went as far as Gulf Point, down in a little below Barnegat Inlet, and as far north as Mantoloking, but we found no red tide above Palms River at that time. Actually, there was, if you see a dotted line up there, it was just a little bit of it. It was reported by somebody else on August 26th. Red tides are special conditions, but they do get your attention. And I don't know of any that have occurred in Barnegat Bay in the last decade or 2. Maybe that's a sign of some improvement in the estuary. But these things are triggered by all the bay's extra nutrients and also many unknown organic materials that are in there. Stuff that comes off from the soap we use, stuff from our medicines, stuff from washing clothes, and who knows what else, that's producing compounds that we have no idea what are doing. Sooner or later, red tides get triggered. Another group of plankton was missed by all the plankton nets in history. These were virtually unknown, and they were beginning to be discovered back in the early 1960s. I dealt at the time I was in graduate school in the 60s with a plankton biologist from South Africa, and she was finding these as well. And there was one biologist in the Chesapeake who was finding them. These are cells that people never really understood they were such an important part of the plankton. They're much smaller. This is a millimeter, is of course on a ruler, very, very small. And this little critter is 0.096 millimeters, 96, about 10% of a millimeter. And it's shown here 1,000 magnifications under the microscope. It too has this tube flagella like a Dinoflagellate, but focus next on the real hot months, particularly in Barnegat Bay, and I'll give you the years of 1967 through 1970, the time periods when the plankton were those big peaks during the hot summers. Now notice in the middle is the normal plankton that I was used to, and the ones that I've shown you, the diatoms and most dinoflagellates, but this excludes, it doesn't exclude the microflagellates. If you separate out the microflagellates, they're at the bottom, and they're almost most plankton at that time. But in my microscope, I was finding things that were incredibly small, 10 to 100 times more abundant than the plankton that I was used to looking at. I had no idea what they were at the time. We eventually gave them a name, but they're 10 to 100 times. If you see the scale on the upper illustration in that, That scale is hundreds, thousands, 10,000, and there were up to 10,000 cells in the count. Very, very abundant. And they're extremely small. That's as good a photograph as I could get. That's the tip of a very small diatom in the upper part of the picture. And Barnegat brown tides were this kind of cell. I never really got full identification on them because we didn't have the chemistry and electron microscopy and whatnot that was available in later years. But this one we gave the name Nanochlorus, and it was extremely abundant. Now, this is not good food for shellfish or even for animal plankton. The oysters that eat this stuff pack their stomachs with it. Their filtering mechanism is enough to get it out, but they can't eat it. They can't digest it. So they don't thrive and they don't prosper. So that's a very serious problem. And this is true for many of the organisms in Barnegat Bay. So I'm coming to a conclusion because when we moved to the Chesapeake in 1971, my new wife, who's probably returned from the vet with my cat right now, these ultraplankton cells, these extremely small nanoplankton cells, were strikingly less abundant than they were in Barnegat Bay. It was a very noticeable thing when I began doing microscopy down there. And this said, and it was shown, in fact, that Chesapeake Bay was much clearer and much cleaner at that point than Barnegat Bay was. So that was a warning to me that Barnegat was in trouble. So my point is, if you were able to control the kinds of plankton, you could make Barnegat Bay extremely much better than what it is. But when I left the Brick Township, Upper Ocean County area in 1971, there were about 21,000 people there. When I went to the Chesapeake, the county I lived in, that I still live in, had 21,000 people. Today, we in Southern Maryland are about 97,000 people in this county, but up in Barnegat, the upper part of the bay has like 400,000 people in the summer.
So I have to stop you there. And I realize you don't live here anymore, and and I just need to correct that number. It's actually 600,000 in the winter and a million in the summer. Yeah.
Is that whole Ocean County or what?
Yes.
But not the watershed.
Well, I mean, so most people who live in Ocean County live in the watershed because the outer boundary of the watershed is so rural. Mm-hmm. But yeah, so year round we have at least 600,000, and in the summer we go up to about 1 million.
Oh my God, that's terrifying. What do you do? How can you control the plankton? I'm at your leisure for any kind of questions you might want to add. Can we close out the PowerPoint and make that work? How do I do that?
Yeah, so just
stop share.
Yeah. So
okay.
I kind of liken this to like stomach bacteria, like when your stomach's all out of whack, like the wrong things are taking over and the wrong right? Like there's still plankton, right? That we understand that there's plankton, it's just not quite the right biodiversity to feed the right organisms. Is that a good way to kind of sum it up?
Mm-hmm. Yeah, I think that's pretty reasonable.
It's a really complicated topic.
Yeah, human diarrhea.
Right. So, folks, do you have any questions for Ken? While people are thinking, I'll share. Margaret was sharing in the chat, and I don't know if you can see this. Your mention of Menhaden reminds her of when she was a kid, probably in the late 40s, when Menhaden invaded Kinsey Cove and couldn't get out again and died there. She can remember the stink. Don't know how anyone got rid of them. Also, you were reminding her of marine biology class in San Francisco in the early 70s, of course. So just a little.
Well, to add to Margaret's insight, when they turned the power plant off to clean the tubes down at Oyster Creek, the Menhaden thought that Oyster Creek was warm water where they could spend the winter, and all those suckers died. Also, one of the curses of Upper Barnegat Bay was the shipworm that ate docks and whatnot, and they loved it in the effluent canal at Oyster Creek. And ultimately, all those docks had to be purchased by the power plant, and it became a canal, an effluent canal, period. So
Mary Finch, I saw you unmuted, or you came back on video. Did you have a question for Kent? No? Okay. Anthony from, who's actually one of our grant students. He is, oh, did you have a question, Mary?
No, I just want to say it was excellent presentation. That's why we came back on. We really enjoyed it. Thank you so much for sharing such a wealth
well, thank you very much. I hope it worked okay for you. I appreciate it. In your beautiful library, actually,
Well,
I couldn't make the computer work in my library, so you didn't see all my ship models and things like that.
well, thank
or
you
the
so much.
decades of journals that I kept while I was in Barnegat Bay.
It was like reading a novel, like a whole group of books. I mean, what you have just presented to us is quite spectacular. Thank you.
I made it all up too. Thank you very much. I appreciate that. So Anthony had a question or something.
Yeah, so Anthony is one of Dr. Paul Bologna's students. For those of you who don't know, Dr. Paul Bologna is the jellyfish scientist who works at Montclair State University. And Anthony this summer will be working with Save Barnegat Bay and Montclair State and Dr. Paul Bologna to study sea nettles, who, if you caught in Kent's presentation, are impacted by the kinds of plankton that they have to eat. And so he's asking specifically which types of nutrients / chemicals cause the most intense plankton blooms.
We don't really know. We thought that it was phosphorus originally in Florida. That was looked at extensively because there was runoff from agricultural pursuits in the Gulf of Mexico. And that didn't seem to be it. And nitrogen, of course, is a role. They can't live at all without nitrogen and phosphorus, but it doesn't seem to trigger blooms. And I have to be suspicious that it's some of these organic materials that are coming in, that they're micronutrients that get them or whatever. The ones in Chesapeake Bay are massive blooms. I mean, there must be hundreds of tons of plankton, even with their tiny size. Quite an amazing thing. And the part of Bay I live in, we don't get it. But we get a winter red tide. Well, Dinoflagellate is very common in Barnegat Bay, Chlorocentrum micans, and it blooms in the winter, both in the Annapolis area and the upper Chesapeake Bay. It turns the water in January, February, most years, red. And we also had a bloom in the Potomac River, which came certainly in concert with effluent from the major wastewater treatment plants up around Washington, D.C. That bloom occurred in February, and it sure looked like nitrogen because they removed phosphorus from the power plant with great efficiency, but they didn't remove nitrogen until maybe 10, 12 years, well, probably 20 years ago now. But those blooms used to occur because the nitrogen that wasn't removed in the upper river was transmitted to the lower river and was able to feed the dinoflagellate bloom. It's not simple, and there's a tremendous amount of research going on. The thing to search is HABs, hazardous algal blooms, and there's tremendous amount of research that exceeds me. I've been administrator for too many years to really, really dive into that. And many of my colleagues are experts. So look that one up, Anthony, see what you get.
DEP has been doing a lot of outreach about harmful algal blooms due to the outbreaks that happened in lakes, freshwater lakes in North Jersey. So there's a lot of outreach information. So it shouldn't be too hard to find.
Well, they're freshwater now. That's very often a phosphorus problem.
Right.
And once you get phosphorus in a lake, and the algal blooms really proliferate, the bottom of the lake can become anoxic. If not the entire water column, then the bottom sediments become anoxic. That releases phosphorus and nitrogen. You feed the next bloom. They grow, they die, settle out, feed the next bloom. It's a very serious problem. And it can make water bodies untenable. We've had that in the upper Chesapeake Bay with some blue-green algae that are normally the ones that cause problems in freshwater.
So no. It's a very complex issue, and it's really interesting. I'm sure we could go. There's so many angles. Chuck, if you want to unmute yourself, do you have a question?
Yeah, I have two questions actually. One is, what do you think is the net effect of the closure of the nuclear power plant over the next several years? And the other, as far as the watershed goes, regulating or banning fertilizers being sold and utilized in the watershed, do you think that would, what would that effect be?
Fertilizer is really important. I'm not completely sure. Oyster Creek and Forked River ought to go back to be relatively natural streams, but you know, the watershed of Oyster Creek was beautiful cedar swamps. My best picture of cedar swamp was when the idiots cut down all those cedar trees. And the effect of the plant was mixed because they used chlorine to sterilize the interior of the tubes and stop worms and bugs and clams and algae and whatnot from growing on them. And it was also a factor of temperature. And first 7 years of my professional career was studying the effect of temperature on phytoplankton. And they killed them. They did a pretty good job. I saw this on not a really widespread scale, but certainly detectable in Barnegat Bay. In the Chesapeake, they were plankton killers. They had canals that were a mile long, and everything in those canals died because the water was like 90-some degrees. So I don't know what the effect on Barnegat Bay is going to be. The benthos is being studied, the organisms on the bottom. And I think that Nancy's company, my wife, actually is involved with some of that with Rutgers University. So that'll come out over the years. But, you know, there's so many other things going on in Barnegat Bay that makes it pretty hard to sort that out. And as Grace had said, 1 million people, for God's sake. I don't know what to do. My solution for the Chesapeake, and in the bureaucratic meetings I was stuck in, I drew a cartoon which solved the problem of the Chesapeake Bay. One was these big buses that go directly to Montana and North Dakota, one-way trip free, picking the people out of the basin. And the second one is machines going around filling dump trucks with impervious surfaces, you know, and pieces of white parking lot lines and, you know, arrows were coming on these trucks. And that is probably the final solution for Barnegat Bay. And I don't think we're going to get people to live. Thank God for the pine barrens. They are one of the last wonderful resources, being chipped away at the very edges all the time. But
well,
I don't know. They're trying to do the McStedeconk watershed now.
Well, don't lose hope. Save Barnegat Bay and others are working really hard to help teach municipalities and folks about what we call pervious surfaces like native plant gardens and rain gardens, green infrastructure, things like that that are just as effective and can help us still have the city infrastructure that we need to live here, but also, you know, infiltrate that water so that it doesn't roll into the bay full of nutrients.
Yeah.
So I have Willie in the chat. Willie, do you want to just unmute and say hi and ask your question?
Sure. Ken, I think you said that there's less silica, starts to be less silica dissolved in the water coming into the bay in the late winter. And
no, by spring, when the river flows go down, the silica is reduced. And you can see it in the diatoms in a microscope. You can actually see them. Their shells are very weak, and the frustules that surround them, they're very thin, and sometimes they're broken open. But go ahead.
So I'm just curious, was that different back before there were a lot of people around here?
No, in fact, you had a cleaner bay in the summer, and that's one reason you probably had more subordinatic vegetation. Back in DeVries' time, when they sailed into the bay for the first time in 1616, I bet they had a lot of SAV underneath them.
And I have a question about Menhaden. Back when there were more Menhaden around, more in the bay, were they importing silica, or were they importing nutrients, or were they exporting them, would you say, from the bay?
Well, I think they're probably exporting. Migratory fish did that. And the herring coming back into the bay, they wouldn't necessarily bring silica back with them, but them migrating up all the streams of Barnegat Bay. And there was a fisherman in my time who fished the Mantaloking, right across from your place. Howard Rogers, he was a herring fisherman. And those fish take nutrients back into the system. In the far west, the salmon were abundant enough and the large amounts of organics were high enough that you could actually get isotopic traces from the open ocean in the forest, in the trees, and in the bears that fed on the salmon. And that, to me, it's documented in the Chesapeake as well from migratory fish, and I bet you could find it in the Chesapeake. But there's signatures, isotopic signatures from the ocean that the fish as adults were feeding out in the open sea. When they come back up, spawn, and die, they leave those nutrients behind. That was really cool to me. It's one of the most interesting things in my life as a plankton biologist.
That is just completely incredible. And it just, I mean, I know those things, but just to hear the evidence of how to describe that to someone, we feel like the ocean is disconnected from anyone who doesn't live on top of it, but it's just all connected.
Yeah, it's amazing.
And
I could
incredible.
see plankton I sampled for a year. I sampled both the bay and the ocean when I was learning about what those luminescent organisms were. And I would find plankton in the ocean earlier in February when that bloom started, and I would find the same ones later on at Mantaloking in Barnegat Bay. And there was a couple of week lag for the plankton, even though the tide came and went every day. So it was a fascinating time. It was a wonderful experience to be able to do my graduate work there. I also did, for those, I'm sorry, the guy who's your intern, I did a paper on the luminescent jellyfish, Nemiopsis, that you find swimming in the bay. And that was really cool. That was a wonderful. I actually took girls on dates in my boat and we went out to pull the plankton net.
Oh my gosh. Okay, wait. So for those of you who don't call it Nemiopsis, the jellyfish that Kent's talking about, it's actually called a comb jelly. And it's not a true jellyfish. It doesn't have tentacles. It's the one you can play with in your hand, and it kind of has like rainbow colored cilia inside of it, and it glows in the dark if you see it in the water. Yeah, those are cool. The comb jellies are very cool. I could see them start as babies. They were the size of the tip of your pinky. Yeah.
And they're called, I've lost the word. Anyway, they were baby.
Is it miroplankton?
Miroplankton are the critters that live on the bottom of the bay.
Okay.
And also on the surfaces of docks and whatever, that produce larvae that are released into the water. And the comb jelly ate the dickens out of that miroplankton with all the baby worms and all the baby clams and whatnot. They would really suck them down. And I could see this in my data from one week to another. It was really pretty cool. It wasn't any brain science. A couple of nets and a couple of bottles, and that was the research project.
Well, so that's the really big important thing to know about the sea nettles is that they eat the miroplankton, and that they are just decimating our crabs and our oysters and our clams, and all these species that are supposed to grow up to be adult versions can't get to that point because the sea nettles are just going at it. I do want to ask. I know you may not have the answer to this, but you might be able to point us in the right direction. But Bill's asking if there's a comparative study of plankton populations before and after the nuclear power plant shut down.
I don't know what was done after my work, but my dissertation showed the same species, but it showed an effect, a limited spatial effect around the effluent of the power plant. And later plants studied in the Chesapeake, it was considered like being a large predator of plankton. that plankton would come in and it would be killed and then discharged. And Harold Haskin at Rutgers said that he felt that what I saw, the effects that I saw in Barnegat Bay, were temporary and that the materials would be metabolized by other organisms. But depends on which ones you kill. The soft-bodied ones died more easily than the hard-bodied ones. So that's actually, people in Barnegat Bay have done plankton work after me, but my dissertation looked at 3 or 4 years before the power plant and a year after. But the basic pattern was pretty similar in the overall plankton. The flow coming in Barnegat Bay is big. I mean, it's tremendous, and it would dilute anything from the power plant. So that's where I came away with them.
So I believe that Dr. Paul Bologna studies plankton, among other things, obviously jellyfish being part of that whole process. But I'm kind of hoping to invite Paul to speak soon. Maybe not right now because it's the field season, but like in the fall when things slow down, to talk about what he's currently finding to do with plankton in Barnegat Bay.
Well, his methods would have to be similar to mine in order to make a real comparison. We did what are called whole bottle samples because of all the tiny, tiny things that we were missing. We would take a half a liter of water in a Nalgene non-reactive plastic bottle. We would add a chemical to it that would instantly kill the plankton before they could really decompose or break up and whatnot. So they were allowed to settle in this jar for a period of a week. And then out of the bottom, we would siphon the plankton and analyze that. So we looked at all the plankton was in a half liter of water. And I followed that for decades in the Chesapeake as well. And we were able to look at extremely small plankton, but not in a live state. They were all fixed by chemicals. And
So
we used a chemical that was a modified Lugol solution, Lugol. It's based on iodine, soluble iodine.
So Anthony might be able to speak to that actually, based on he's working in his lab.
Yeah, I don't know what Paul's been doing with plankton. What do you say, Anthony?
Where'd you go?
He slipped off his sleep.
No, he has a question here in the chat, and I think I know the answer to this. But the baby comb jellies, are they a phyra or only the cnidarian species? So just to translate for you folks,
they're not a phyra. No,
right?
They're named after another oceanic ctenophore, and I had it in my mind while you were asking the question. Pleurobrachia-like organism. And they have long tentacles that hang down on the side as opposed to the combs that are the normal capturing process. And they look like pleurobrachia. They're ovoid and they're the smallest ones I've seen were in a dip plankton sample. And they were smaller than the tip of your pinky. Maybe, I don't know, I don't know, 3 or 4 millimeters maybe. And they came up very quickly. That was one of the most remarkable things because they were chomping down that miroplankton. And of course, the ctenophore, the Crisiora, they also eat miroplankton and they eat the Nemiopsis. And some work in the Chesapeake said that they were actually helping the survival of oyster larvae, the Crisiora. I think Denise Brightberg was one of the, maybe Anthony can look her up. She was a colleague of mine for a few years in the Bay. And she just retired from the Smithsonian.
You know what Paul was saying? Paul Bologna was saying that that the interaction between comb jellies and sea nettles in the Chesapeake is completely flipped from the interaction between the comb jellies and the sea nettles in Barnegat Bay. And even in northern Barnegat Bay versus southern Barnegat Bay, the sea nettle interaction with comb jelly is flipped. So in the northern part of Barnegat Bay, the sea nettles are like top dog. Then in the southern part of Barnegat Bay, we've got comb jellies are kind of, they're high up on the list, but not as like, not the sea nettles are not nearly as abundant. And then in the Chesapeake, it's actually flipped around. The comb jellies are like, it's just really interesting how in different ecosystems, the same exact organisms have different behavior, like ranks.
Comb jellies can survive all winter in the Chesapeake when you don't have a really survive. And there's some big comb jellies down here in the wintertime. And they're around. They haven't actually come back in numbers yet in Chesapeake Bay, which estuary do I live in. But when the comb jellies show up, that means the sea nettles are soon to follow, and then I can't swim anymore. So I feel these things as I'm swimming. I was out there today. I thought I hit one yesterday. It was a piece of grass, and I can swim until the damn sea nettles come, and they're miserable. That to me was a tragedy in Barnegat Bay, just a tragedy. Because I don't like to get stung, and I don't want to swim in that. So.
Most people feel similar, and Save Barnegat Bay is working on a project for the next 3 years to try and lower the population of sea nettles in Barnegat Bay. So we have, it's no small task, but we're going to be trying to scrub bulkheads to try and eliminate the ephira.
All right.
The plastic bulkheads are where Paul Bologna found that they were growing.
Well, Nancy has found the polyps of my wife is a benthic ecologist, and she's found polyps on things as small as a large sand grain.
Yeah.
So I truly support any effort to get rid of them. You know, Congress actually made a law against sea nettles at one time. They passed a law that was supposed to outlaw sea nettles and useless.
I would
Paul
love a law
and maybe Anthony are probably familiar with the work of David G. Cargo, who was an old friend in the Chesapeake. He's the first one who paid attention to nettles. The colonial literature has no references to sea nettles. I don't know whether they was there or whether people never swam. The Indians swam. You think they might have made references. You don't see the references until the Civil War. And there's a prison camp about 30 miles south of us on Chesapeake Bay, where they reported there was one of the guys who ended up in Binghamton, New York, in a prison keeping a journal. And the blue crabs show up, and the sea nettles show up. He said they start about the size of a nickel, and they grow up the size of a small dessert plate. And they're big. I don't know if you ever see the bigger ones in Barnegat Bay. It seems like they're smaller, and there's some genetic difference. I've read this, but I don't know.
Yes.
That's Paul's field.
You're right. Yeah, so we could go on for hours about sea nettles and other things, and obviously we need to have more presentations because we had enough interest tonight and last time that these are things people want to know about. So we'll definitely make that a point in the future. So I just want to thank you again for being here tonight and for sharing your breadth of knowledge. I mean, it's wonderful, and it just sparked all of our curiosity to continue learning about these things. So if anyone has any questions as they're milling around all this information, please send them over to us at Save Barnegat Bay, and we'll get them to Kent or email them or call us or anything that you have questions about, because I know it's a lot and it's a weird topic because you can't really see plankton as well as you can see other things. So
get a red tide, you'll see plenty.
That's true, but we don't want those. So no. We don't want a red tide. So thank you again, Kent, for going through everything to be here.
Thank you for listening.
Yeah, thank you so much. And you're getting lots of applause. All right, this will go up on YouTube, and we will see you next time.
Thank you very much.
Thanks a lot, Kent.
Good night, my friends.
Bye.